Hello, can we start then?
Yes.
Hello, good night, because we are in Spain now in Europe and it's very late. It's 8:30 p.m. So I started working at 8:30 a.m. So it's very late. So this is the session of FENStatS, which is the Federation of National Statistical Societies in Europe. And we selected 3 European researchers representing different European statistical societies based on in Norway, Poland, and Spain, that will present some of their ongoing research in statistical modeling within the academia. The 3 researchers are at different stages in their careers. So SARA MARTINO is from Norway, and she will be talking first. Then we will have a PhD student, which is Dorota, which is now in the States doing one of his research stays. And then finally, we'll have a speaker from Spain, which is very close to my office, JAIONE ETXEBERRIA. So let's start. Sarah, you have about 25 minutes for your session. Thank you. Thank you to the organizers also.
Thank you very much, and thank you for inviting me to this very nice event. So I'm going to talk about a joint work with several people in Rome, and this is a very applied work, so there will be not so many equations in my presentation. So the question that we are trying to answer is, where are the dolphins? So my colleagues in Rome, they are all working with dolphins, and they are very interested in understanding where they are and where they are not, in order to try to keep them wealthy and to avoid a lot of disturbance from humans. So in particular, we are interested in this area. This is an area you see here is Rome. This is the coast of Italy. So this is a part of the Mediterranean area. And it's a very interesting area for dolphins because there are a lot of small islands. There are very steep, the sea is very deep down here and not very deep here. So it's a very, very interesting area. And in particular, we are interested in these 2 species of dolphins. Now, I never remember who is who, but one is a species that lives very close to the coastline, while the other one likes to live where the water is very deep and colder. So how do we try to estimate where these dolphins are? Well, traditionally, what researchers have done is to either sit on the ferries. So here you see the ferries that goes from Rome to Sicily and from Rome to Sardinia. So they sit on top here of the ferries and look out and record whenever they see a dolphin. The other alternative is that some researchers, they have their own boat and they leave from the harbor in Rome and try to go around and search for these dolphins. So as you can imagine, we can only observe dolphins where we go. So we will never observe a dolphin up here, for example, because we have just not been there. So the thing that is important to notice is that the observation process introduces a bias in our data. But the interesting thing is that we know the searching protocol. We know how long these researchers have been outside. We know exactly when and where they have been. So this means that there are protocols and statistical models that allow us to correct for such a bias. One of the traditional way of doing this is this transect, where we imagine that we are on this boat, on this transect, and then we can see that the probability of spotting a dolphin decreases with the distance from this line where the ferry has gone. So we can put this into the model and we can correct for this. But what is the problem with this data? Well, this transect data cover very small space and time domain. So we only see dolphins in a very, very small part of the domain we are interested in. And the other thing is that they are quite costly to collect. I mean, we need to have people that sit on these ferries or on this boat for quite a long time, and they are there looking out and recording whether they see something or not. So, I mean, it's not the most, the cheaper data that we could get. And also they are very sparse. So then the question is, do we have more data source that we could use? Are there other things that we could use in order to understand where these dolphins are, and can we model this jointly with this traditional data in order to have a better and a more robust model? Well, so my colleagues in Rome, they found out that actually in this part of the Mediterranean, there is a lot of boat trafficking. Mostly it's people with leisure boat, so sailor boat, that go out from the harbor in Rome, and whenever the weather is nice, they go out with their boat. And of course, when they see dolphins, they take pictures, because I mean, it's a very special thing that you are there and you see a dolphin. And when they take a picture, they often put these pictures on social media. So mostly they've looked at Twitter, Facebook, and Instagram. And although I have the old Twitter logo, this that now is called X. So what this researcher in Rome have done is to contact all the people that have put this picture on social media. And on the picture now, you also have the location of, so you have latitude and longitude of where the picture has been taken. So they have collected all this data. So this is quite a lot of work. But it turns out that these are a lot of data. So if you look at these two pictures now, these are the two species of dolphins, the bottlenose and the striped dolphins. And here we see that the red points are the data collected on ferries. And here you see the trajectories of the ferries that I've showed you before. It's very clear where the ferries has gone. And this UNI-RM, these are the dolphins that have been spotted by scientists on their own boat. So are these black dots. But you see that the great majority of dots here are these social media data. So they are a very big data source. I mean, it's not huge, but it's big compared to what we usually have in this field. And so they could be, in theory, very valuable, very, very valuable. But you see that also in these green spots, there is a bias, because most of these green spots here are located, this is at the harbor in Rome, and these are islands that are very, very nice. So a lot of people go there with their boats. So you see that if we would just use this data, the estimate we would get, there would be biased towards the places where people go, not towards the places where, I mean, that we would, our result would be like where the people are, not where the dolphins are. So then our problem is that we have these three different data sets. They are all so-called presence-only data, because of course we know where the dolphin is, but we don't know where the dolphin is not. So each of these three data sets is biased in its own way, and we only know the searching protocol for two of them. So for two of them, we have ways of correcting this. This is fine. But for the third one, we don't have information. And this is the largest database. And this is something that is getting very common now, that the largest data set is the one with most bias. And we want to include it in our model, but we don't want this large but biased data set to destroy all the other information from the other ones. So our goal is to do a joint model where these three data sets work together, but where for each of them we account for its own bias. So this is our goal. And then I'm going to talk a bit about which statistical tools we are going to use. And mainly we are going to use log Gaussian Cox processes. And in particular, we are going to look at so-called thinned point process, where we use a detection function to model where the process has been observed and when it has not been observed. So these are models that are continuous in space. So when the space becomes bigger and the data set becomes bigger, we of course run into computational problems. And in addition, we have these three models together, so the computation becomes even more heavy. So the computational tool that we are going to use is that we are going to represent these continuous random fields on space with this SPDE representation, which gives us a formulation that is very computationally efficient, because it's a Markov random field. So it has a sparse precision matrix. And to do inference, we are going to use integrated nested Laplace approximation, which is a tool to do approximate Bayesian inference for latent Gaussian models, which has become very popular in the last maybe 15 years. So just very in brief, what is a log Gaussian Cox process? Well, in a pi process, what our observations are the locations of the points. So imagine we have a space and we observe all these points, and these are our data. So we have n points on a domain, and we say that this is a point process that is governed by some intensity, lambda s, which is what we want to recover. So this intensity will be high in areas like this, where there are a lot of points, and will be low where there are no many points. So we can write the likelihood function, which will look like this. And what is special about this log Gaussian Cox process is that we have another level of randomness, where we say that this intensity function here is not is also random. So we say that the log of this function is a Gaussian process. So now we have two levels of randomness. And the thing is that these processes are not analytically tractable, so we have to approximate them somehow. And the traditional way of doing this is to divide this into a grid and to count how many points you have in each grid point. But we are going to use another approach, which is this SPDE approach. This is based on representing the space, so the domain that we are interested in, to divide it instead of using a grid, using a mesh, that is this triangularization. And then we are going to approximate this Gaussian random field with this formula here, where these phi here are basis function that are known basis function, and these z I here, they are some Gaussian weights. And it turns out that this function here, that this thing can be well used to approximate a Gaussian process. And the most important thing from the computational point of view is that this vector of weights is going to have a Markov properties. So it means that it's going to have a sparse precision matrix, which in turn makes computations a lot easier. The other thing that makes this kind of mode of representation very good for point processes is that now we don't need to approximate. I mean, we are approximating the Gaussian field, but we don't need to approximate the location of each point. We can use it as it is. So this is also another advantage compared to this grid that was common before. Our next tool is this thinned point process. So what is a thinned point process? So imagine that we have this, this is our intensity, that is what we want to recover. And these are the points that we observe if we could observe the whole process. So if we could observe all the points, then this would be our data. Now this is in 1D, but you can imagine the same in 2D. But the point is that we cannot, we are not able to observe all these points, but we are only able to observe each point according to this thinning function. So this is the probability of seeing one of these points if it is there. So in conclusion, what we observe is this here. So we observe the product of lambda and g, and these are the points we observe. And what we want is from this point to reconstruct the intensity function. Of course, we need to have information about this, otherwise we are lost. So just some few words about what is INLA. INLA, as I said, is a computational tool for doing approximate Bayesian inference for this special class of latent Gaussian model. And in particular, we require that this latent Gaussian model have some Markov properties. So the thing is, to make it short, that all the things that I've mentioned before can be framed into the framework of latent Gaussian models. So integrated nested Laplace approximation can be used for all these models. And the other nice thing is that INLA comes with R package. There are two now. One is called INLA and one is called INLABrew. And INLABrew is just a wrapper around INLA. And so it means that they are also useful. I mean, they are available also to people that are not computational statisticians, like, for example, the people working with the dolphins. Okay, but let's look a bit more in details how we have modeled this, this problem here. So we imagine that the true, so the unthinned density, here I should have a log, sorry, is given by some intercept, and then the effect of some covariates, which are the slope of the bottom of the sea and the depth and the water temperature. And this is, while this US, this is a Gaussian mark random field. So these are continuous, it's a Gaussian field, sorry, that we represent through a Markov random field. So this is a continuous spatial field. So this U here could be also space-time. And we wanted, was our first goal, but the data are very sparse in time. So we end up with only a spatial model. But this is the observed intensity. This is the true intensity. But to get the observed intensity, we have to multiply this true intensity by the detection probability. So what we have at the end in our model is that we have three observed intensities that share one part, which is the true intensity, and then each has its own thinning function. So then the question is, how do we model? So we have seen how we model the true intensity, but how do we model the thinning function? So for this ferry data, we used a very classical method, which is this decaying, so saying that the probability of observing something decays with the distance from the transect. So as this form. For this researcher data, then they said that they are very good control on dolphins up on a certain distance, and they don't see anything below, above that. So this is how we modeled this second thinning probability. And then the problematic one is the social media, right? Because we don't have information about I mean, we don't know where people are. We don't know how long they've been outside. We don't know anything about these small leisure boats. So what do we do here? And we tried, so our assumption is that you have more sightings, the more leisure boats you have. So in places where there is more dense leisure boats, we expect to have more dolphin sightings. And in places where there are no boats, then we don't see dolphins. The problem again is that we don't have data about these leisure boats. They are not recorded, they are not tracked, so we don't know. So then we tried three different things. The first is to say that, okay, close to the coastline, we expect that there are more boats. The closer to the coastline you are, the more boats you are expected to find. The second one is to get some data about boat density from this EMODnet. This EMODnet is a platform that registers, I mean, every large boat has to be equipped with something that is called AAS, which registers where the boat is. And all this information are recorded in this EMODnet data. And the last idea was to use animal sightings intensity as a proxy for a small boat. And I'm going to say something more about that. So the thing is that we don't observe, I mean, this Facebook and this social media data, are not only about these two dolphins that we are interested in, but people take pictures of a lot of marine mammals. So because these points here are all the sightings that have been recorded on social media. And the point is, because these marine mammals, they have very different habits. So some live very close to the coast, some live very far to the coast. Then our working hypothesis is that this estimated intensity here reflects the boat intensity and not the animal intensity. And we have good reason for thinking that this is true. For example, here, this is the Rome harbor, which has a very high boat density. These are these islands that are very popular, that also have very high boat islands. This is the Isola del Giglio, that is also very common. And this is the route that people that want to go sailing to Sardinia take. So in a way, what we do is that we use all this extra information in order to try to estimate the boat intensity. So when we put things together, then our model looks like this. We have the true intensity that depends on some covariate and a Gaussian random field. And then we have 3 observed intensity that share one part, and then each has its own special part. And then what we have tried is to see, okay, what happens if we do not model the, so if we say that this G social, so the detection part of the social data is constant, or if it's based on the distance from the coastline, the boat intensity, and the sightings intensity. So one question, because here we have this product here. And then one question, of course, is this model identifiable? Well, the answer is that, because, I mean, the thing is that if you have low sightings, this can either be because there are no animals in the area, really, it's not a good habitat, or because there are no observers in this area. So this is a problem of identifiability. We take a Bayesian approach. So the way we face this problem is to gather information about the observation process and to use a slightly informative prior to guide the inference in order to not to have this identifiability problem. So if we look at the results that we get here, I'm just going to talk about one of the dolphins, the one that live close to the coast. So this is the posterior median of the intensity surface that we get if we ignore this thinning process. And you see that there are a lot of dolphins in the area in this result, but this is mostly where the harbor is. So this is where a lot of boats are, not a lot of dolphins. This is the result if you use the distance from the coastline, and it doesn't seem to help a lot. I mean, we still kind of have exactly the same pattern. This is if we use this EMODNet. And at first we thought that this was a very good idea to use this EMODNet, but then we realized that most of the data that we collect, they come from small boats that don't have this AIS. So then the small boats, they don't behave like these big boats that are recorded in this data set. So then at the end, we ended up with choosing this animal intensity as a proxy for the small boats. And the result that we got, the ecologists were very happy about, because what they say is that these dolphins is almost uniformly distributed along the coastline. And this is what we get. And even this, I mean, that is a bit higher around this harbor, this is not so strange, because, I mean, on the harbor, there are a lot of sailing boats and fishing boats that these dolphins like to follow. And the nice thing about our model, of course, is that we also get an estimate of the posterior distribution of the variance. And this is the CV, because this is an exponential model. So the standard deviation is high where the mean is high. And then you see that the variances is smaller close to the coastline, of course. And this is the model that we chose, which is also the one that has lowest variance somehow. So what do we conclude here? That this is a very complex problem, but it's something that in a way is very topical and is becoming more and more common to have these problems where you want to mix different data sets that are all biased, and then you have one that is bigger than the others and maybe even more biased, but you want still to use this information. What we managed to do in this model is to model several data sources jointly. And this, the researcher, they had no idea how to do before. And we can correct for the bias that is induced by each of these observational processes. We can also recover, I have not talked about this, but we can recover some known covariance effect. And then we can also estimate the intensity surface with the associated uncertainty. One point that I want to make is that, I mean, these two, especially if you use this framework of Inla and Labrousse, you can fit very, very complex model and you get an answer, whatever you do. The problem is that a lot of these complex models, they are maybe slightly undetermined in a way that you have to be very, very careful with your priors and with your choice in order to have a meaningful result. So I think I am perfectly on 25 minutes.
Yes, thank you very much, Sarah. Very, very interesting talk. I want to ask you a question.
Yes.
What about if you don't use the social media data and you just use the other two sources of data? Which are your results?
Then you get something that, I mean, it depends which one of the dolphin you look at, because where are the data? For one of the dolphins, so for this one, the striped dolphins, it kind of works because you have a lot of observation here along these transects. So you get some meaningful inference. But for these other ones, they live close to the coast, so you almost don't see them from the ferries. You see there are very, very little information from the ferries, and you get some from the boats, from the researcher boats, but they are only located here. So it's very hard to get the inference that is spatially meaningful.
I see. I understand.
So I think, I mean, you really need to use these extra data sources in order to do some inference in space.
Yeah. Can you treat the two species simultaneously in a single model? Because you say you analyze separately, right?
Yeah. So we use them together in the detection part. But yes, you can also do it together. Yes, we haven't done it because, I mean, there are not so many data, although it looks like a lot of points, but this area is very big. So there are not so many points, and we wanted to keep things as simple as possible. But yes, I think you can do it.
Mm-hmm. And now one final question is, when you use INLA and INLA-BRU, you are using the traditional Laplace approximations, or you are using variational base?
I'm using the variational base now.
Okay, so you're using the compact mode.
Yes.
Yeah, otherwise...
But I've done it with both. I mean, the result is the same.
Okay, so it's possible also to use the traditional way of fitting
yes.
INLA.
Yes.
I see. Then thank you very much, Sarah. It's very, very interesting. I wonder if you published already this work, or...
Yeah, this is published, yes.
It is published? Where it is published? Yes. Which is the journal?
It's on ecography.
Ecography, I see. Congratulations. Yeah. Very, very interesting.
Thank you.
Thank you. Thank you.
Thank you.
So now we have our next speaker, which is Dorota. Dorota, to me, is very difficult to pronounce your first name.
Don't worry.
Okay, it's difficult. I chose you because you are originally from Poland, and then even though you are now in Barcelona, I wanted to have different countries representing Europe. So it's your turn, okay? Thank you very much for accepting to be here today.
Thank you very much, Lola, for introducing me. My surname is MLYNARCZYK. Yeah, it's like Polish surname, so yeah, it is quite difficult to pronounce, I can imagine. Okay, so thank you very much in general for inviting me to this event. It is a pleasure to be with all of you today. So just a second. Okay. Okay, so I'm in the final year of my PhD studies, and since its beginning, I've been focusing on statistical models for biodosimetry. So today I would like to provide you with a brief explanation of this topic. So first, let me talk about radiation in general. So it is everywhere, every day. Did you know that, for example, bananas are a source of radiation as they contain a radioactive isotope potassium-40, which emits beta radiation? Bananas are also used to translate the dose of radioactivity to the number of bananas which should be eaten to receive the equivalent radiation dose. This is even called banana dose. So, for instance, eating 50 bananas is equivalent to the dose received in a dental X-ray. Spending 1 hour close to the reactor core shortly after the explosion in Chernobyl was estimated to be more than 300 million bananas. Unfortunately, it was also 6 times more than the lethal dose. So definitely by eating 1 banana, don't be worried, you won't start to glow in the dark, okay? You can eat bananas. But the estimation of doses which are fatal and so on took humanity more than 100 years since the discovery of radioactivity. So during this time, like really crazy things happened. Okay, here is an example. In the 50s in the United States, a toy called Atomic Energy Lab was sold in shops for about $50. And this kit, it was like for children, included real radioactive material, such as a small amount of uranium ore. As it was advertised, it allowed children to explore the principles of nuclear physics and atomic energy, but like for me, it is quite surprising, and I think for all of us. So after another 50 years, this particular toy, it made it onto the list of the 10 most dangerous toys of all the time. Okay, but fortunately, after years of exploring radiation, safety systems have significantly improved. Nowadays, things are much more under control, but still occasional minor radiation incidents sometimes happen. So here you have a map of Europe, and you can see the locations of radiation events over the past 50 years. So this radiation incident took place in locations such as ports, refineries, or hospitals. Like, because there are many applications of the nuclear technologies around the world, especially in energy production, in food processing, and in medicine. So sometimes something goes wrong. Several websites are available on the internet where you can check the details of the radiation incidents, and you can also find the nuclear event report. So here you have an example of such a report. So a worker destroyed the electrical cabling system of a radiation protection cabinet, and the safety system didn't work correctly. So this worker was exposed to X-rays for about 30 minutes, and what is also important, he didn't wear a dosimeter. This means that the dose which he received was unknown, but in order to provide him with the best possible medical care, the radiation dose should be estimated. So as you can see on the slide, the resulting dose was determined by use of biological dosimetry. So biodosimetry refers to the use of various biological markers to quantify the radiation exposure of individuals suspected of being exposed. So in a possible scenario, an accident occurs and samples from several patients are collected and transported to the nearest laboratory. Then the samples are analyzed and the results are communicated to the medical staff who make treatment decision on this basis, based on these findings, right? So in a small scale accidents, only few samples need to be tested. Like in the example of this worker, just one sample needs to be analyzed, but in large-scale accidents, a significant number of samples must be investigated. Therefore, the dose estimation process must be like quick, simple, and efficient. Okay. Okay. So several biomarkers can be used for dose estimation, but the choice of a specific biomarker depends on this scenario under consideration. So I work with biomarkers, like in general, I work with biomarkers found in blood cells. Blood samples are generally the most studied as they are like easy to obtain. And in emergency situation, also like all medical professional know how to draw some, blood, right? So it is like easy to use in emergency situation. So today I will discuss the modeling of one specific biomarker, which is called chromosomal translocations. So if a cell is exposed to ionizing radiation, chromosomes inside the cells can break, and the broken ends may rejoin incurrently in different patterns from the original arrangement. Using some biological techniques, this may be seen under a microscope at metaphase, a stage during a cell division process. So here you can see examples of the photos taken during the analysis, during the scoring. So it is a human karyotype, so the cell has 23 pairs of chromosomes, 46 chromosomes in total. And on the left side, you can see that 3 chromosomes, not chromosomes, like pairs of chromosomes are painted, numbers 1, 2, and 4. And on the left side, this is the situation when everything goes well and chromosomes do not break. But on the right, you can see that chromosome number 4 has some like unpainted fragments, and these painted fragments is attached somewhere else. And this is exactly translocation. Okay, the situation when the chromosomes break and we see like the chromosomes which has painted and unpainted fragments. Okay, so in something what is important, that the translocations are stable over time. So this biomarker, for example, was used to study the irradiation levels of populations living in like next to the Techa River. Techa River is a river in Russia where the Mayak nuclear complex spilled millions of cubic meters of radioactive material during the Cold War. But the inhabitants who were living like close to this river were unaware of the threat, and that's why the estimation process happened many years later. But still, it was possible to estimate doses because translocations remain like on stable levels after like many years of the radiation accidents. Okay. So, to simulate the data from real scenarios, blood samples from healthy donors are irradiated under controlled conditions in a laboratory. Then biologists with whom we collaborate, they analyze these blood samples cell by cell, and they record the number of translocations in each cell. So it is important to note that radiation doses in this part are known, okay? So this data is referred to as a calibration data, and our aim is to fit a statistical model to this data and create a calibration curve. As we have some counts, like the obvious choice and the most used approach in biodosimetry is a Poisson model. So we have just one random variable, and we assume that it follows Poisson distribution. But the mean number of aberrations in cells can be also modeled somehow. So for the translocation assays, the most used approach is to model lambda as a linear or quadratic function of dose. Okay, so this is the basic scenario with basic data, and here you have an example of such data. So in the table, we see that we have like data for 7 different doses. Like dose 0 means that the cells are not irradiated, but this is something like translocation are also sometimes occur in cells like just naturally, okay? So for example, for dose 0, 7 translocations were found and so on. But in general, we see that hundreds of cells were analyzed, and for each radiation dose, we know the number of translocations found in cells. In the plot, you can see the fitted calibration curve. So in general, higher doses in radiation result in more aberrations in cells. So this is like quite obvious. And the next step, the most interesting step, is the estimation of the dose. So imagine that we have a new blood sample coming from potentially irradiated person, and we analyze this blood sample cell by cell, and we have also like the number of translocations found and so on. But in this part, the radiation dose is unknown, and our goal is to estimate. So, well, let's suppose that we found 53 translocations in 500 cells, so it gives us a mean of around 0.1. And we can see on the plot that the corresponding dose is a bit higher than 2 grays. But as we are statisticians, we are not very satisfied with this type of result, right? So let me specify the model in a better way, okay? This was just an example how it works, but now I just want to specify the model, and because I am working mostly within Bayesian approach, it will be a Bayesian model, okay? So the likelihood is defined according to the model. So here I use an example of the Poisson distribution, but sometimes we use other distribution to model this data, okay? This is like the most common, the most simple one, but sometimes, for example, the mixture of Poisson distribution is used, or negative binomial, or any other distributions, okay? So, so we have here the likelihood, and well, the parameter lambda is defined using the quadratic function of dose. And for estimation, the capital letter D is the dose which we want to estimate, right? So in total, we have like 3 regression coefficients, the parameters of the model, plus the unknown dose which we want to estimate, okay? And for each parameter, we need to choose prior distributions. For the regression coefficients, we decided to use non-informative priors over a positive range, because like the mean number of aberrations should be positive. But here we come to the advantage of the Bayesian approach in dose estimation in process and in general in biological dosimetry, because the prior for dose we can choose according to some knowledge about the radiation accident. So, for example, if a patient has some symptoms shortly after the accident, we know that the dose was very, very high, that he received like significant radiation. And we can choose prior, for example, on the range starting on 1 gray, right, or above. And that's the advantages of Bayesian approach in this case. And well, to get the posterior distribution of the model's parameter, we generally use MCMC methods. As, for example, for this model, the full conditional distributions of the parameters given the rest of the parameters can be clearly defined. We usually use a Gibbs sampling method, because it is like quite easy to apply in this case. But our primary interest lies in the dose estimation. So we want to know something about the dose D. So what we are looking for the marginal distribution of dose. So this can be obtained by integrating the posterior distribution over the rest of the parameters. And based on this estimation of the dose, the medical professionals can decide how to help to the irradiated person. Fortunately, thanks to the safety systems nowadays, in most of radiation accidents, the doses are very, very low. So the hospitalization is not even needed. But as I said before, after a large-scale accident, radiation doses, like the estimation of radiation doses would be necessary for triage to determine the allocation of treatment and doctors. Okay, so here you have an example of the result, the marginal posterior dose density. As we use the data coming from the laboratory, we know that the real dose was 2 grays. It is marked by a red line in the plot, and the mean of the posterior distribution is around 2.1. So model works quite well, but we hope it would be better, that the result would be better with more calibration data. Okay, we just have like the 7 calibration points. Maybe with more calibration points, the model would give us better result, but still we are quite okay with the results. Okay, so this was the basic scenario, and to complicate things a bit, now I want to explain the partial body radiation scenario. So imagine that only a part of a body was irradiated, and like the other parts of the body were sheltered from radiation. So this is, for example, the case in targeted radiotherapy. And another example of the accident that like a few years ago in a Polish hospital, the machine for radiotherapy was broken, and the 5 patients received much higher doses, like during radiotherapy, than intended. Okay, so it was they just had radiation burns into their skin, and the dose estimation was needed, but it was only like part of the body was irradiated because it was targeted radiotherapy. So it means that this is the scenario under consideration. Okay, so biologists simulate this scenario by mixing irradiated blood samples with non-irradiated blood. This means that many cells in our data do not exhibit aberration as they were not irradiated. So we have the excess of 0. To account for this, we use a 0 inflated Poisson model. In addition to the existing parameters, we introduce one more parameter, parameter omega, which specifies the proportion of irradiated cells in the samples. So in other words, 1 minus omega is the proportion of non-irradiated cells, which accounts for the excess of zeros. Okay, so omega also can be modeled using some covariates. But now I want to give you the example that in biodosimetry, the research question isn't always about the dose estimation. There are also other scenarios under considerations. And since we collaborate a lot with biologists, they sometimes ask us very interesting questions. So allow me to provide you with an example of such a question. So many cells are damaged by significant radiation, making them impossible to be analyzed. So initially, the samples were mixed in a certain proportion of irradiated and not irradiated blood, but then this proportion has changed because during the analysis, many irradiated cells are excluded because they are like completely destroyed. Hence, the research question becomes, what is the actual proportion at the analysis like of irradiated cells, right? At the time of the analysis. Okay, so the survival rate in the literature, it is described as a decreasing exponential function of the dose. We can do some like modifications and we can find the expression for the actual proportion of the irradiated cells in the samples. Okay, this enters to the model as the parameter omega of the zero inflated Poisson distribution. So the key point is that omega depends on the dose and the initial proportion, the proportion which is assumed. And here we have new parameter gamma, and this parameter is called survival index, and it needs to be estimated. Okay, just it needs to be estimated. So here you have an example of the results. So this is the posterior distribution of the survival index gamma in this model. But for me, the more informative is the table, which is just below. So this table gives us the actual proportion of irradiated cells based on the dose and the initial proportion of irradiated cells. So when for example, we take the highest dose in the data set, 5 grays, and initial proportion of irradiated cells was 0.5, it means that the equal mix of the irradiated and not irradiated blood, then the actual proportion is 0.25. So like 0.25 of irradiated cells and 0.75 on non-irradiated cells. So this, like, the proportion change has changed a lot. So if you are interested in this topic, there are some references, and well, thank you very much for your attention, and happy International Day of Women in Statistics and Data Science for all of us.
Thank you. Thank you. Very good. Very interesting and very, very difficult problem, because you need to work very close to biologists and all these people to understand well this problem, no?
Yeah, exactly.
Can you please tell me, how do you validate your results? For example, the last results you showed that the initial radiation was the initial proportion of irradiated cells was 0.5, but then finally you get this 0.25. How do you validate that? How do you know that this is a nice data solution?
So in this case, we can't validate it as we do it using the dose estimation process, where when we know the exact dose. But here we know that this is in accordance with the other studies on this topic. And like the gamma, which was estimated previously in other studies, it is like in the same interval as our result, and that's how we can validate it.
Okay, I see. I'm interested also in the first map you show us with different colors, which is why the different colors? What does it mean? Why is the blue or the yellow or the red there?
Yeah, probably, I'm not sure about that, but probably there is a scale of the radiation events, and well, some of them are not dangerous at all, and some of them they are like very dangerous, not all for the people, like in general for public, right? So this is like the radiation scale of accidents.
I see. You started your working with Pere Puig, as I know, but he's not Bayesian, or he is now.
He is now
because why you changed to the Bayesian?
No. So first I worked with Carmen Armero, and she's completely Bayesian.
Yes,
and
I know.
Virgilio Gomez Rubio. And I have 3 directors of my thesis. Like
I see.
Carmen, Virgilio, y Pere. So we are discussing a lot.
Yeah. So you find the Bayesian inference more appropriate here in this problem?
Yeah, exactly, because we can, for example, as I say during the presentation, we can choose the prior distribution of those accordingly to the accident. And this is like good advantage of using Bayesian approach in this case, right? Because we know something about accidents, and we also have like some calibration curves, not only one, and we can also put the priors for the regression coefficients of the calibration curves as well, some knowledge which we have before, and well.
Yeah, I agree. That's a very important point. So you are now in the States, right? And
yeah.
You are in a center of radiation, or in a research center, or just in the university?
No, I'm at the University of Nevada in Reno. And well, I'm not doing here nothing connected to radiation, just statistics.
I see. So you are doing other things. Yeah. I see. So Dorota, thank you very much. I am very pleased that we were able to connect the four of us, because it was not easy, and that's why we are so late in the European Eastern time. It's almost 9:30 p.m. So thank you very much. I think that we can go on with JAIONE, and if we finish a bit earlier, that would be nice. Thank you.
Hi, good evening, everyone. Thank you, Lola, for inviting me to this special day and session. So, yes, so I would like to present what is meant to be a dissemination talk entitled Unveiling Spatiotemporal Patterns of Rare Cancers, Multivariate Bayesian Models with Adaptable Shared Interaction. And this is a joint work with Garasi Reddy and Lola Huarte from the Public University of Navarra in North Spain. So first of all, I would like to say that this work is conducted within the context of disease mapping, which is a specific field within spatial statistics that focuses on the analysis of real data in an epidemiological context. Real data, you also know as lattice data, is a type of statistical data that represent information in aggregated or summaries within a predefined geographic unit. So we would like to tackle 2 goals in this work. First of all, we would like to enhance the accuracy of estimations for incidence and mortality of rare cancers at both country level or a smaller administrative unit. We would like to also provide predictions in areas where cancer incidence data are unavailable. So just to start a little bit of the background, you all know that cancer is a very important disease affecting millions of people worldwide, and health authorities are highly interested in monitoring or controlling. So to do so, they take into account, they consider the most updated new cancer cases, the incident cases, and deaths that take place yearly in a country or in a smaller administrative unit. In most of the countries of Europe, it's important to say that both data types, or incidence and mortality, usually come from different data sources. In most of the European countries, the mortality data are collected by the national statistical offices, providing them at national level, at province, state, municipality, census tract, or even divided by age group or sex. On the other hand, the incidence data are collected by population-based cancer registries that there are usually within a country many registries located in different parts of the geography, covering different proportions of the population. Here, in that context, excluding Germany, Nordic countries, and some others, in the rest of Europe, the net of the registries do not cover the entire population. As, for example, here in Spain, this is the administrative division of Spain by provinces, And here in gray, you can see the provinces in which we do have a cancer registry. The net of these registries cover approximately the 30% of the Spanish population. And although they are high-quality data sources, they can only provide a partial picture of what is going on with the incidence. This is a very common problem in Southern Europe, in most of the countries in the Southern Europe. So in the literature, there are many different statistical procedures that are proposed to estimate incidence in countries not entirely covered by the net of the registries. The most common approach to solve this problem is to try, simply saying, is to try to model or try to compute the incidence to mortality ratio observed in the pooled registries, and using the national mortality, try to extrapolate the incidence at the national level. There are specific versions of that approach, and the most important thing is that this is the one used by the Global Cancer Observatory, Global CAN, that is the most important organism in cancer research worldwide. So we think that this method could be a little bit restrictive because they assume that the incidence to mortality ratio observed in the pooled registries is a good estimator of the national one. A feasible solution for that is maybe consider methods that account for possible regional spatial variability. And we think that maybe multivariate models to analyze jointly mortality and incidence or some concept type could be a good solution. So to do this, to replicate, to analyze the models we proposed, we need a complete data set, a country in which the incidence and mortality is collected for all the states. And this is the case of Germany. The German Center of Cancer Registry kindly provided us different incidence and mortality data and the corresponding population at risk. And they precisely provide us breast, lung, and prostate cancer data for the 16 federated states of Germany, divided by age groups, and for different time periods for incidence and mortality. So the first goal is to see how these multivariate models estimate or predict incidence, cancer incidence in regions where the incidence is not registered. So, and the second goal is to extend this into a space-time context. So the first objective to deal with this, we need first to do a descriptive analysis. Here you have the crude incidence rates, and in the bottom side, the crude mortality rates for lung, prostate in the center, and breast cancer. This descriptive analysis revealed that we need to handle with two important aspects. First of all, the high to low lethal. We have high to low lethality cancers. For example, depending on how close the incidence and mortality rates are between them. For example, lung cancer is a high lethality cancer because the incidence and mortality rates are very close to each other. Otherwise, in the case of prostate or breast, these are a low lethality cancer, right? On the other hand, we can see, for example, in the case of lung or breast cancer, there are high similarities in the spatial patterns, right? Here you have the northwestern regions are the highest in incidence and the same in mortality. In breast cancer happens more or less the same, and for prostate cancer, at first glance, these two patterns seems different, right? So if we consider this aspect, we follow the next step in order to emulate the Spanish situation. Here we have Germany, and for example, we remove the incidence data for stage 2, and then in this case, we have the 90 to 80% of the population covered by a registry. Little by little, we remove the data of incidence data for some regions up to this context. In all these situations, we keep the mortality known for all the states. Then, in step 2, the modeling context will be a disease mapping usual context in which conditional on rates, we assume that the observed incidence and mortality cases follow a Poisson distribution, right? So in this context, our interest resides in modeling jointly the log incidence and mortality rates using different multivariate approaches. The first approach could be used the well-known shared component models. We have here 3 proposals in which a disease-specific intercept is considered. Then we have the shared component term in which delta is the scaling parameter, and then we can add some other spatially unstructured random effects. That could be individual or outcome specific. The other proposal is to use a model. In that case, the log incidence and mortality are modeled as a disease-specific intercept, plus these thetas are a collection of random effects whose joint distribution specifies how the dependence within diseases and between diseases is defined. More precisely, this theta could be defined, expressed as phi times m, where phi is a matrix of non-zero or zero mean Gaussian random effects composed of stochastically independent columns that usually follow a spatially correlated distribution, such as intrinsic curve, proper curve, etc. And M is a non-singular matrix that induces dependence between the different health outcomes. The audience is referred to Botella Rocamora for more detail. So, using this approach, we use the model fitting inference and validation. Is used as SARA said, with the well-known integrated nested Laplace approximations using the R-in-lab package. And in order to compare the results of the observed versus the predicted incidence data, we use different measures such as absolute relative bias, mean absolute relative bias, or the Davis-Sebastiani score, in which the expected and the standard deviation of the posterior distribution of counts are involved. So the first results indicate that at 98% of the population covered, both models are equally competitive, but as the percentage of the population covered decreases, the shared component model provides better results. In that case, the lethality of the cancer is not important, but the more or less similarities in a spatial pattern is it, right? In that case, the results for lung or breast are a little bit better than for prostate cancer. It's true that using the German data, the number of areas is small, so we try to increase the eukaryote simulation study, increasing the number of areas, and see what happens if we do, let's say, the same process. In that case, we can conclude also that in all the scenarios, the shared component model is still good, providing good results. So now the objective is to try to estimate the incidence at national level. It means to estimate the incidence counts at this level. So to do so, we try to compute the posterior mirror and the quantiles of the posterior distribution of the national incidence count. To do so, we follow some steps. First of all, we got some samples for the joint posterior distribution of the models in order to get the posterior marginal distribution of the linear predictor, the log rate. Using the exponential function, we get the posterior marginal distribution of the rates in each area, and then using the Poisson distribution, we finally compute the posterior marginal distribution of the counts in each area, and finally we did the sum. In that case, we see that both models provide quite good results. It's true that the shared component model is a little bit better because it provides more precise estimates with narrower credible intervals. It seems that in the case of prostate cancer, the M model provides more accurate posterior means, but wider credible intervals. Sorry. So But for us, the most important results is the following. If we compare the results, our results, with the ones provided by the incidence to mortality method used by Globocan, we are reducing substantially the relative errors achieved by these methods. In this case, we can just could obtain the results at 50% of the population covered. There are no more validation studies in that case, but it's true that we are improving the results substantially. So just to say, this work was recently accepted for publication in the Journal of the Royal Statistical Society, Series A. So let's see what happens if we try to extend this into a space-time context. In here, additional issues are need to solve. We still have some portion of the population that is not covered by a registry. On the other hand, we see that not all the registries are established in the same year. So we find incomplete data series or some underreported problems for some registries. The third additional problem is that due to the complexity of data collection, classification, validation, and processing, cancer incidence data become available with a delay of 2 or 3 years compared with the calendar year. So our goals in that context is to try to predict incidence in areas without or underreported incidence time series, and provide also short-term cancer incidence predictions. The natural extension is to think in models in which the log incidence and mortality is modeled jointly using a disease-specific intercept, this shared component term, and adding to them some time-specific random effect and maybe some kind of interactions, the one proposed by Norhealth. We saw at first glance, using some rare cancer, these models don't work well, so we need to think in other proposals. So our new approach or a new model is to try to propose spatial temporal models with flexible shared interactions. That means that in that case, the interaction is a shared term in which some rows are shared between these two random effects. These rows could be flexible. I mean, we can choose a different row for each time specific, for each time calendar time, or we just consider the same row for the whole time period, or maybe something in between. We can choose a suitable number of scaling parameters that can be adjusted based on the data under study. So with this approach, we implement these models using ILNA. We need to solve also sum to zero. We need to impose sum to zero constraints also to ensure the identifiability of the different terms and the scaling parameters. And we use more or less the same validation criteria in order to see how this model predicts versus the survey data. So in that case, we used, let's say, a more rich data set, the Great Britain data set, in which pancreatic and leukemia cancers were considered. Those are very rare cancers, divided in 142 council areas, divided also by groups, gender, and considered in this period, 2002, 2019. So we in the first step, we fitted these models, the model with the independent shared space-time interaction and the one with the shared space-time interaction. The first results indicate that the model with shared interactions provide better results in terms of DIC, YIC, and logarithmic score. We can then represent the spatial, the temporal, and the interaction term. But the most important thing is see how this new model predicts. So the first results indicate that this model predicts pretty well comparing with the model including dependent interactions, because we are reducing also slightly the bias, the error, and the Davis-Sebastiani score. So the discussion, just to end, we think that this novel proposal is interesting. It's useful for our proposal and for cancer research, but we need still do some simulation study to comprehensively assess the strengths and the weaknesses of a new approach. And we need also to compare our findings with other alternatives, for example, that are specifically designed to address underreporting or missing data in a space-time context. And that's all. Just to end up, I highly recommend you to visit us in our webpage, Spatial Statistics Research Group webpage. And that's all. Thank you very much, everyone. Thank you, Lola, for inviting me.
Thank you, JAIONE. You were very quick. It seems that you would like to go home because it's just 15 minutes to 10 p.m. and you have to drive. So I'm not going to ask you any questions because I am involved in this work. So I just simply give you thanks for this nice presentation. Also to the three speakers, also to SARA and Dorota. I'm very happy to share this session tonight. And well, we just celebrate this nice day. And hopefully next year we will be here also celebrating again the International Day of Women in Statistics and Data Science. So thank you for the organizers, and that's all for our session.